Right, and this leads us to the second lecture, which will be focused on the function and structure of the small intestine. So you will be able, after this lecture, hopefully, to describe the anatomical features of the small intestine, describe the cellular function and organization, especially of the epithelia in the small intestine, talk about the motility and the mechanical digestion which occurs in the small intestine, and the secretions in the small intestine. Of course, also describe the structure, the role, and the secretion, both endocrine and exocrine, of the pancreas. So the main function of the small intestine is to complete the digestion and carry out the absorption of nutrients. This is facilitated by the secretions that change the pH of the chyme to the optimal condition for the intestinal juices to work, because the chyme is too acidic. So, the bicarbonate secreted into the small intestine will allow the pancreatic enzymes and the other enzymes which work in the small intestine to work at their optimal pH, which is close to neutrality. And the overall, occurrence, in the small intestine is that macromolecules, macronutrients, starches, lipids, proteins get broken down into their monomers. Okay, so proteins get broken down into amino acids. Complex carbohydrates such as starch get broken down to monosaccharides, simple carbohydrates, while fats, triglycerides, get broken down into fatty acids and glycerol. And then in the small intestine, they get absorbed via the portal vein or via the lymphatic vessels. So the anatomy of the small intestine is a pipe of 6-7 meters with a diameter of around 4 centimeters, which is more or less half of the diameter of the large intestine, which is around 8 centimeters. It's divided in 3 regions: the duodenum, which is the part downstream of the pilar attached to the stomach, is around 0.3 meters long. The jejunum, which is between the duodenum and the ileum, which is 2.5 meters long. And finally, the ileum, where most of the absorption takes place, which is the terminal part of the small intestine, which is around 3.6 meters long. And it's connected via the ileocecal sphincter to the large intestine. So at a histological level, what we do observe in the small intestine are 3 modifications of the mucosa. Okay, we do have the permanent folds called plicae circulares, which are, the overall folds of the mucosa in the small intestine. Then we do have within the plicae circulares finger-like projections inside of the small intestine called villi. And then the epithelial surface of the villi and of the overall intestine is characterized by epithelial cells with an apical membrane which presents protrusions of the plasma membrane known as microvilli or brush border. So you can see overall how the generation of these folds, from the plicae to the villi to the microvilli, allows the overall increase of the surface to volume ratio, to increase to a maximum the absorptive surface while keeping the volume relatively small. So the fold number for the plicae circulares is the highest in the jejunum, a little bit less in the ileum, and the lowest in the duodenum, while the villi in length are the longest in the jejunum, a little bit less in the duodenum, and then finally the shortest in the ileum. And as you can see on the villi, we do have different subpopulations of epithelial cells. The villus absorptive cell, the goblet cell, the enteric endocrine cell, stem progenitor cells, Paneth cells, and undifferentiated crypt cells. So, this is a typical columnar epithelium, which is mostly responsible for the absorption of nutrients and electrolytes, so monosaccharides, amino acids, and fatty acids. Crypt cells are actually also involved in secretion. And these epithelium, like any other epithelia, is constantly renewing, and this in particular is self-renewing. So the villus absorptive cells, are the main contributors to the brush border, and they're usually on top of the villus. Goblet cells mostly secrete mucin, which is a protein, which is a major component of mucus. Enteroendocrine cells are endocrine cells, as it says. So they produce hormones that then get secreted directly into the plasma, and these are mostly cholecystokinin, gastrin, secretin, and glucose-dependent insulinotropic factor, or GDI. And the stem progenitor cell, which is, a stem cell that can differentiate in any of the epithelial cells of the small intestine villi. The Paneth cell, which is mostly involved in the secretion of antimicrobial peptides and proteins, such as lysozyme, because, the pH content of the small intestine is compatible with the growth of bacteria, but we do not want many bacteria in the small intestine, as we will see when we talk about the microbiota. So in the stomach is easy because the pH doesn't allow bacteria to grow, but in the small intestine, if we don't have these enzymes, it may be colonized by bacteria that then would interfere with the digestive and absorptive processes. Different story for the large intestine. And then ultimately the undifferentiated crypt cell, which can proliferate to replace lost enterocyte. So the exocrine secretions that we have in the small intestine are mostly mucus produced by goblet cell and forming a thick layer that protects the mucosa against the damaging action of acids and proteases. Sorry. And it's mostly produced in the duodenal portion. Water, a lot of water gets secreted in there, and it's mostly produced in the small intestine, mostly produced by cells in the crypt, and water combined with mucin makes the mucus runnier, and this is good for the movement of the content of the small intestine. And also water is it sounds like a stupid thing to say, but without water the enzymes don't work. So we need to have the adequate amount of water to allow the digestive enzymes to carry out their actions. And finally, the bicarbonate, mostly produced by Brunner glands, which release mucus-rich bicarbonate secretions. And this is very useful for protecting against the acidic action of chyme leaving the stomach, and it also generates those more alkaline conditions where the pancreatic enzymes or the digestive enzymes in general, which act in the small intestine, can actually work at. So in terms of endocrine secretions, cholecystokinin is produced by eye cells in duodenum and jejunum. Secretin is produced in S cells overall the small intestine. Somatostatin is mostly produced in the D cells, which are both in the stomach and in the duodenum. And finally, the gastric inhibitory peptide, which is mostly produced by the K cells in duodenum and jejunum. So the presence of fat in the food promotes the activation of enteroendocrine cells, which produce cholecystokinin, which then in turn stimulates the exocrine cells of the pancreas to secrete digestive enzymes. It also, cholecystokinin leads to the contraction of the gallbladder, which contains bile salts, which are needed for the emulsification of fats. And it also promotes the opening of the sphincter of Oddi to allow the bile salts to reach the small intestine. So at that point, the fat contained in food is emulsified in micelles, increasing then the surface to volume ratio of the fat that can be attacked by the pancreatic lipases. And so you can see how cholecystokinin stimulates both the secretion of bile acids and pancreatic lipases in order to digest the fats. The hydrochloric acid also, stimulates the enteroendocrine cells in order to promote the secretion of secretin, and secretin itself leads to the secretion of bicarbonate from the pancreas and from the liver in order to neutralize the acidic pH of the chyme entering the duodenum.